resistance, microbial forensics, microbiota, and health and hospital-based water sanitation, hygiene interventions, which we share in the listing. Christine George, who was the lead on that author, was here visiting last year. Yeah. So it's wonderful to have you here. We're looking forward to hearing from you. I can tell you that Dr. Munir is really open for collaboration. He's just such a sort of an engaging and warm scientist, and so we're really thrilled to have you, and we look forward to hearing from you. So thank you so much.
Thank you, Dr. Pamani. This is an incredible introduction. I don't know whether I deserve. It is really a pleasure, honor for me to be here, to be able to talk in front of you. Basically, this is an unusual time. I was traveling to India before I came here, and I was presenting this talk in India. The exact title is there because I didn't have any opportunity to change this, because my computer that I carried is an old one. Basically, my fellow colleague bought one MacBook Pro for me, but I found that very difficult for me because I'm used to using Dell. And also before that, I had a Sony Vaio, which I lost in Amsterdam. If you travel to Amsterdam, be careful. My bag was stolen. Actually, I saw someone was running with my bag. I just tried to go after him, but I just turning back. I just saw that another person was nearing my other bag, so I had to give up. So, and then I started using Dell, and this Dell computer was broken, basically. This was broken because I was using this in flight. So the person sitting in my front seat just pushed back his seat and broke my computer. But still, I just managed to use this. This is what should I say? Your obedient pet, like this. So all the talks, everything that I do, I have this in my computer. So fortunately, I had this small thumb drive that I could bring this talk to you. So this is also an unusual thumb drive because I don't use my, regular thumb drive for transferring files to other computers. So this is just for one-time use actually. So the one that I used here is one-time use thumb drive. So when I go to the meetings, I use my talk, I carry my talk with this and then I drop it. So this is going to be of so use to me today. So my computer was unable to be opened actually. So I tried to recharge, but it was not opening. So I had planned to, I was attending SKMH meeting in Baltimore, and then I had planned to go to Chicago to attend another meeting. If you are familiar with Review 2018-17 meeting. So unfortunately I had a battle in my kid, and I have to go back from here and then back to Bangladesh because I have my mails and my pending work accumulating to a level that I will have to go because I cannot work here. So anyway, I want to just share with you informally what we do. Dr. Pavani has already mentioned that our work is ranging from environmental to clinical to antimicrobial resistance to microbiota to forensics to comparative genomics. So the reason is that like in the current era, you cannot do one thing without the others actually. So these are linked. So when I started working in the field of environmental microbiology back in the early 90s, basically I started isolating bacteria from water and see their toxin carriage, their drug resistance profiles. In those days, there was no means for us at icddr to go for isolating plasmid, unfortunately, in those days actually. So I tried because I had a friend working in Japan, so he helped me to isolate DNA from bacteria, Vibrio cholerae. So I also analyzed the water for different chemicals like nitrate, phosphate, sulfate, silicate. These were done by titrimetric method. As a chemist, Diana, you must be familiar. Maybe these days you don't hear about. So in those days, I did titrimetric detection of all these chemicals to see what chemicals or what environmental factors actually trigger the bacteria called Vibrio cholerae that causes cholera actually. So the cholera bacteria has a very interesting feature that the cholera bacteria is difficult to be isolated from environment, but the bacteria is survives in the water. And when there is an outbreak, you can go and collect water and you can isolate the bacteria. But as soon as the outbreak finishes, you cannot isolate the bacteria because the bacteria enter into a non-culturable state, or called viable but non-culturable state, VVNC, or unculturable state. So this is a way of bacteria to be kind of escaping from different, kind of environmental things. So and then I did my that was my master's work. I was doing my master's thesis. So also I detected the Vibrio cholerae bacteria by immunochemical method called fluorescent monoclonal antibody detection method. Specific monoclonal antibody level doing fluorescent dye helped me to detect the Vibrio cholerae bacteria. So I don't have unfortunately a slide to show you. Also, we detected Vibrio cholerae bacteria by molecular method. So that was very limited actually. That was very limited in those days. Then I did my postdoc, PhD in Japan. That was on the molecular pathogenesis of Vibrio. Basically, I tried to find out some of the factors that bacteria Vibrio cholerae uses to adhere to the gut of human. So as you know that for bacteria to cause diarrhea, they need to adhere to your, if you swallow them, they have to have the capability to bind to your intestinal mucosa or villi or lining of those villi to be able to secrete toxin to cause the disease. So I identified a couple of factors that plays role in adherence. I produced antibody in animal model, also in a mouse model, a rabbit model, and found that those antibodies specific to those factors actually had the capacity to inhibit the adherence of the bacteria. So that was published in Infection Immunity, a couple of other journals in America back in those like probably 1996, 97, around that time. Then I did my postdoc in National Institute of Infectious Disease, Tokyo. That was on the signaling. I'm just taking 3-4 minutes to cover that full, how I am master of everything but jack of none and anything maybe. So that was on the signaling. So the signaling means the regulation of virulence gene expression in the bacteria. So I actually knocked down or mutated or actually nullified genes in the genome, as you know, that regulates everything, the genome, the gene, DNA that we have. So I just knocked down each of the DNA and see, I just try to identify genes that actually have the regulatory function. So then what I did from the online gene bank, I just decided sequence. I just found, I did first of all, I identified from my study that which are the genes that regulate the expression, and then I just prepared segment of DNA and just cloned it and tried to revert the mutated gene to the bacteria to see the reversion of the expression. So this was done in a manner that you will be surprised. This is like manually doing everything. So I worked day and night. I slept in the laboratory. In Japan, if you go there, you will see that there are beds, portable beds, blankets in every laboratory. You can sleep there. You can buy your food, go to the vending machine, buy coffee, and just sleep the night. I slept. My wife used to carry dinner for me at time. We used to share some time, and then I used to go back to my work. So this is how I work actually. Nowadays, you can do this volume of work by just maybe 4 hours in a day. The ones that I did taking like 12 hours, 16 hours, 18 hours, anyway. Then I came back and joined Dhaka University microbiology department. I was there actually from the beginning. I was a lecturer there, I was assistant professor, then I was made associate professor. So when I was associate professor, then icddr colleagues, I was in University of Maryland for postdoc, I was in Germany for postdoc. Those are the things that I actually had linked. So the University of Maryland and Johns Hopkins group was having a project in icddr. And basically since I was doing my master's in one of the projects, So they were my kind of master supervisor, PhD supervisors involved in this. So they called me and I came and I got stuck. I was unable to go back to Dhaka University. So that's how I became involved with so much of things actually. So my work in signaling now actually helped me to understand the behavior of the bacteria, the bacterial community that actually are in the environment, also in the human body, how they are actually interacting with each other and playing an important role, and how the chemicals that actually we have in water, it could be antibiotic, it could be recalcitrant chemical, it could be pesticide, it could be like fertilizers or whatever, has got tremendous influence on the microbial community. It is just like sometimes the chemicals dictate the microbes. Sometimes the microbes actually has any direct impact on the chemicals actually basically. So this is how the in the environment it is an intricate, very, very delicate thing that is happening in the environment. So I was wondering at any point, like at every stage of my research, that what are the things that is happening in nature. So if I collect some water from one place, I get bacteria which is sensitive, but around the same time, if I collect water from other place, I get the same bacteria which is resistant. I have papers published in that in some journals. You can see the same bacteria actually from different places has different profiles of antibiotic resistance. So that actually triggered me to wonder what is actually is playing with all this actually. So this I would rather go. I will take 15 to 20 minutes. Is it okay? If you have any time, is it okay? Okay. So I have not included any slide to show you the aquatic environment, but 1 liter of water, ocean water, can have more than 1 billion of bacteria. It includes bacteria, virus, microorganisms. 1 liter equivalent soil can have the similar number of more than billion number of bacteria. So these bacteria actually from environment, we play with soil. When I was a child, I used to play with soil. I used to mouth soil. Soil mouthing is very common everywhere actually. So that's how the bacteria, the newborn actually gets the bacterial community through soil mouthing actually, and drinking water. And fingers are always like, the children puts fingers into mouth. So they play with toys and everything. This is how we get them. And during childhood, you probably will not find a person who didn't have diarrhea. It could be watery diarrhea, it could be mild to moderate diarrhea. So severe watery diarrhea are caused by some bacteria which are notorious, like Vibrio cholerae, cholera-causing bacteria, Ecology bacteria, rotavirus, and some parasites. They cause such watery severe diarrhea which needs to be treated. But most of the time, the mild sub-infectious or moderately dehydrating diarrhea are nothing to be worried about. So they go silently and we don't care about them. Children has loose stool, mother is feeding, child is playful, so nothing to worry. So this is how the environmental bacteria gets into our body, and they cause, of course, not all of the bacteria swallowed by individual causes disease. Only the susceptible. Okay, so only the susceptible individuals develop infection, and they recover, and they also infect. They can also seed others, like person-to-person transmission, fecal-oral mode of transmission. They can go back to the soil again and then come back to the. So it is German scientist Metagenomics. He actually proposed that actually human intestine is a very important place for microorganisms to colonize. So this actually gave rise to an area of important research, and you can see from these that those bacteria actually colonize our body. They have preference, and they preferentially colonize our body, and depending on the substrates, it could be chemical, it could be polysaccharide, food that we eat are shared by them. So what food we eat decides the type of microbiota. If I drink alcohol, if I drink something, if I drink milk, if I don't drink alcohol, so these are the factors. If I eat rice, if I eat burgers, fast food, Kentucky Fried Chicken, these are the factors that play a very important role in deciding. And you can see from here, there are ligands in the human body, like our mucosa, intestinal mucosa has specific receptors, like you have blood groups different. So the A, B, O, different blood group holders have different receptors in their body. So different bacteria can bind to their body. And this bacteria binding, actually I will show you the difference, can make the difference. Like your behavior, your personality, everything is decided by them. Because the human body has the third organ, which was ignored, now called the third or neglected organ, which as a human being we neglected, but now we find that this is our third organ. Okay, so they actually bind to different level. Like this, I will show you in the next slide. See, the body has different organs and different substrates, different food, and different kind of factors that actually invite the microorganism, allows the microorganism to colonize and settle there. So the mouth is the entry point of the gut. So mouth actually cavity houses more than 100 different species. So the skin can have millions, and the gut and the intestine can have 100 trillions of bacteria. So it is enormous number, even more than our body cell. So it is basically 100, even more times more than our body DNA, the microbial DNA that we have in our body. So as you go down from mouth to up to the colon, up to the anus, as you go down, the number of organisms increases. And here you can see 10 to the power 7 in the ileum, 10 to the power 12 in the colon. Sorry. And I can touch, yeah. So this number is 10 to the power 12. So this bacteria starts colonizing our body as soon as we are born, and this colonization continues in the first year, and there is enough diversity that is required to be established for a child to be healthy. So whatever diversity you have, this diversity that is created in 1 year continues throughout your lifetime. So if a wrong bacteria colonizes for some reason, this will continue throughout your life. This will have long-term impact in your life, living, your behavior. So there are lots of papers, like intestinal microbiota in health and disease. Nature, no less than Nature is publishing this. Functions of the gut flora. How many different functions did they? When I was a student, microbiology student, I used to know that the commensal bacteria, they don't do any harm to us, but they live with us. This is a harmony, this is a peaceful, like living together. They don't cause any problem. But later on we came to know that they can be deadly. They can cause disease, they can kill us at times. When our immune system is down, we become sick, we became affected by any other things. They can cause death. So I now believe, before I forget, they are our inbuilt kind of messenger to cause our death. Basically that is the fact. Probably we'll see someday. Oh, so the intestinal mucosa, the lining, are the places that they live, and where there is complex carbohydrate, the human being cannot degrade. But this is the microorganisms that can degrade that for the human and make it available for absorption. So sometimes we are advised by our physicians to go for some food, fibrous food, some prebiotic, probiotic type of food that helps to decide the type of microorganisms. So diet microbiota interactions as moderators of human metabolism. So a microbial perspective of human developmental biology. This is a huge area of research and interest for the modern scientists. So the human body, if you compare, the whole body has the amount of DNA which is here, the red, and the rest of the body is microbes. So this is the comparative view of the amount of DNA. So the amount of DNA that actually comes from microbes are transcribed, translated into proteins, and those proteins actually work inside the human body, nowhere else other than human body. So you can imagine that this enormous amount of DNA translated into protein, and these proteins function in human body. So how much intense influence it can have on the human. So this is what we are trying to understand. So this is an example of diarrhea, cholera. So when cholera bacteria infects human, they cause diarrhea, secretory diarrhea. This is like soapy water. So imagine that this is stool, nothing other than stool. So this stool carries bacteria that are supposed to be colonizing your gut. So basically diarrhea and cholera is washing your body, washing your intestine. So it is not allowing any bacteria to settle down. So this is where we want to concentrate on. So you see, an ill-health mother giving birth to one ill-health child. So why they are ill-health? Because of the frequent diarrhea that they suffer. 3 to 5 episodes of diarrhea. This is something that I don't want to talk about because too much science. So the short-chain fatty acids produced by bacteria, basically they help in absorption. So this is an indicator of the gut microbiota. You can, rather than going for the bacteria, you can measure the short-chain fatty acids like acetate, butyrate, propionate. This is diamycerium. These are the indicator of your gut microbiota. So we just measure the gut short-chain fatty acids in the gut. So when a child was suffering from cholera, we gave him glucose saline, glucose plus arabinose-resistant saline plus rice saline, and we measured the stool volume as well as the short-chain fatty acids. So what we have seen is that when a child is given rice saline, the rice saline works very well, the short-chain fatty acid increases very much, and compared to the other two salines. So that is why rice saline has become so much famous. This work was done back in the, it was done before but published in 2010. So this is an indirect measure. That's why actually the rice saline that you know now is actually something that came out from this kind of research. That actually this rice saline helps. If you have glucose saline, other salines, any like you can go for like sugar, like we call it like small amount of sugar, small amount of saline, salt can serve as a very important rehydration saline. But if you compare, the rice saline is the best. This came out from our research. So when the child is given treatment with antibiotic, when they arrive on the day 0, this is about cholera, okay, cholera children. So you see day 0 profile. Oh, what we do, we isolate DNA from the stool samples to see how many different types of bacteria are there. So this stool DNA is then analyzed by denaturing gradient gel electrophoresis to see the different positions of the bands indicating different flora. So you can see here, this Vibrio cholerae, the cause of cholera is here. This band is cholera bacteria, this is here. On day 0 when the patient comes to the hospital. Can you see this number of bacteria in this cholera child is very, very less. That means I showed you the soapy water-like stool, rice water stool. It causes expulsion of all kinds of bacteria. So you can see the picture of this. So as the antibiotic is given on the day 0, so we isolated DNA from day 1, day 2, day 3. So this is the period we give a dose of antibiotic, 3 days course of azithromycin. So during that period, E. coli bacteria shoots up. And this is called antibiotic guarded E. coli floral restoration. But you see, unfortunately, all other bacteria disappears as the antibiotic actually acts against them. They may be friendly flora, but it's not allowed by the antibiotic to settle. So antibiotic allows only the drug-resistant E. coli bacteria. So as the drug stops, at day 7, this band changes, the prominence of the band changes, so other bacteria starts restoring. Okay? So other bacteria, small bands, you cannot see, but I can see very faint bands because their quantity is very, very less. When they start restoring in the gut, their amount is very, very less. So slowly they restore, but as of day 28, the number is not good enough. So what happens when we culture the bacteria of this 1 month after the recovery? What we see is that the children serves as a reservoir for multi-drug resistant bacteria. So E. coli bacteria that you saw prominent is here, the most abundant multi-drug resistant bacteria in the gut of these children. This was published recently. So this is the picture of how we knowingly or unknowingly inviting multi-drug resistant bacteria to settle in our gut. So this multi-drug resistant bacteria not only cause diarrhea but can cause wasting in the long run. As you know that in our intestine, if pathogenic bacteria can colonize and settle there, they can cause blunting of our villas. So the blunting causes disruption and nutrition absorption is hampered due to this villus blunting. If you go in the internet, you can see villus blunting due to colonization of the intestine with E. coli bacteria. So this is our E. coli bacteria that we saw was prominent when we were using antibiotic. So when we did the sequencing of the gut flora of child at day 0, day 2, day 7, 20, 28. So at day 0, this is Vibrio cholerae bacteria, the cause of cholera. So you see how the antibiotic changes the pattern. At day 2, E. coli. Did you see at day 1 and 2 the E. coli was prominent? Here it is E. coli, EC, E. coli. So this is our sequencing, the whole genome sequencing data, community data actually. So this is the overall floral pattern in cholera children. Okay. So see that this bar shows the overall community, bacterial community, which the diversity is so much important for our healthy living. Can you imagine this? V means what? B means Vibrio cholerae. This is not victory. This V means Vibrio cholerae. So this Vibrio cholerae bacteria and E. coli is like more than 95%, and the rest of the flora is like this, marginal. So imagine that cholera bacteria, when cholera actually recovers, a child recovers from cholera, basically they are in a situation that they need a long kind of intervention to get their flora, appropriate flora. to be healthy. So we have seen floral succession, like at day 0, it is a pattern, and Vibrio cholerae bacteria is up to day 2. As time goes on, they disappear, but there are change in the flora. But there is some common flora. So this actually is very important for us. Yeah, this is very important for us because this will allow us to go for intervention. So this intervention is something that is going to help us. So how to help a child to recover from this kind of problem? What we have to do? We have to stop cholera. We have to We have to do something to stop this epidemic infection, because you saw that one episode of cholera can cause this havoc. So if a child suffers from 3 to 5 episodes of cholera or diarrhea, so it causes a lot of damage. So there is a nature paper shows floral immaturity, which we have shown long ago. Floral immaturity in a child which is 18 months old can have the floral pattern which is like 6 months old child, healthy child can have. So this is called floral immaturity. So this is something that I wanted to share. This is the cholera child. This is the healthy child. Can you see the distinct bending pattern? Number, intensity is very different. So we just analyzed the gut flora of 2 groups of children. One is healthy and one is malnourished. So these are age-matched. Actually, their BMI was different. So these 2 groups of children showed very distinctive patterns. The healthy one had much more diversity than the malnourished children had. So when we identified the bacteria, we saw difference in the healthy and malnourished. The proteobacteria, which are the cause of diarrhea, was less in healthy children, much more in malnourished children. So we have identified 2 distinct groups of flora in healthy and malnourished children. So this bacteroidetes firmicutes are prominent. So you can see in nature papers that shows that bacteroidetes and firmicutes are the 2 bacteria that can help decide your health in a positive way. So if someone is obese, can have this bacteria more than their lean counterparts. And this proportion of bacteroidetes and firmicutes, like if the firmicutes is more than bacteroidetes, you are obese. This is a fact. So the malnourished child had disproportionate amount of proteobacteria here. See, proteobacteria, which is E. coli and others. Anyway, I just wanted to, this is a slide which summarized our data. So the healthy and malnourished children has this distinct fingerprint of community, microbial community. So Klebsiella and E. coli is in this color, and healthy individual had bacteroidetes and firmicutes more prominent, but proteobacteria far less. So this is something that can answer why malnutrition is prevalent in our part of the world. Thank you very much. Dr. Kamani has already mentioned the importance of the intervention. We are having the wash intervention. As I mentioned, that if we cannot stop, if we cannot reduce the number of episodes of diarrhea, we cannot help them. So this is what is going on now. We are having intervention, and this intervention is really helping us. So Dr. Christine was presenting her data. We have been able to reduce cholera by 50% in the intervention arm, in which we have taught them how to make their water safe by putting chlorine tablets or boiling water, and how to wash hands and avoid being infected 5 times a day. We provided them some hardwares like bucket with tap, with a washing station, hand washing station, liquid soap, and this is not a bar soap or very expensive soap, but some powder soap put into a PET bottle, used bottle with a small hole in it, and hang near the toilet, open toilet. So when they come out, they can have some and just wash their hands. We provide them the hand washing station and also drinking water station, 2 different colors. So when you visit Bangladesh, maybe you can see this is really helping. So we are now bringing something to help them, as we know that this is so important. Thank you very much. Yeah.
Thank you so much. It's super interesting. I'm wondering the future. It seems like antibiotics, it wipes out everything. I know in the past, like I've used probiotics to increase the good bacteria. What's the future of treating bacterial disease without antibiotics?
Basically, antibiotic is given not only to help the patient to recover early, to reduce the hospitalization, because cost is involved, because if we could reduce the hospitalization, that we could cut it, we could reduce the cost, as well as when we are giving antibiotics, the causative bacteria can be kind of reduced. The load of the bacteria that is going to the environment is also reduced. So this has got a lot of implication. So that way you can bring down the peak of the epidemic. Like you cannot imagine that how big the peak would be, but by providing antibiotic, you could sense that this will have an impact. This is called an pilferage impact. Like the antibiotic is going to kill the bacteria. It is going to be in the environment to serve as a kind of inhibitor for the bacteria to multiply, to affect more people. So definitely.
I think your point is when you get to a place where the available antibiotics have relatively little effect, then how do we treat bacterial disease?
Yeah, this is basically cholera is, you don't need to give antibiotic. Like if you provide them with oral rehydration saline, if they are in a situation that they cannot swallow anything, you go for intravenous or an ORS. Without antibiotic, they will suffer longer, and this will increase the chance of further contamination, but the patient is not going to die. So even without antibiotic, you can do it. But how do you stop the agricultural use? How do you stop the medical, clinical use? Because this is not only diarrhea that you are treating patient with antibiotic, but there are many other different types of infectious diseases. The doctors are changing antibiotics from one to others. How can you stop it? It is something that is needed to be taken care of by the policymakers from state level.
Yeah, I'm curious, you mentioned that the permanent microbiota kind of happens in the early ages. Is it possible that an adult could completely change the microbiota? Let's say a person migrates from one country to the other, but then permanently live there. Would that microbiota completely change and be permanent?
Basically, when you travel, there are traveler's diarrhea. We sometimes call it a Crohn disease or cytomembranous colitis, or sometimes when you are traveling, maybe Lisa, you are traveling, maybe you have some experience, like you have some diarrhea, you are not killing anyone, but you are having it. Maybe you develop secretory diarrhea, or maybe the consistency of the stool is different. So you can sense that your bowel is not normal, but you are having some kind of alteration because of the change in the ambience, because of the food habit, and the new environment can have new different types of flora. So when they come in and fight with your own flora, there is some change that takes place. But you cannot just eradicate or eliminate, but of course some new flora will be introduced, so that is possible.
So you mentioned proportions of gut bacteria in relation to obesity. So I'm wondering which one is causing the other. Is it behavior that's causing microbiota, or is it microbiota that's causing behavior?
Basically, my training in this signaling, my postdoctoral work in signaling, signal transit, that helped me to understand this kind of small things. Like when the bacteria senses a little bit of kind of change in the environment, they switch off. You know, like you feel like if some cold ice is touched from the backside, someone touches ice in your body, you will sense that. And definitely you will turn around and try to prevent someone from doing that. So the bacteria also senses the minute, very, very tiny, very small signal and can modulate their action. I mean, whatever is needed for them to either shut down their system. They're utilizing something, they're taking up some sugar and other things from the environment, and they are active, moving from one place to another, producing, reproducing. Okay, so they easily can switch off their system, can become dormant, can become non-responsive to normal culturing methods. So what happens when your body has some bacteria transcribing some gene DNA and translating into proteins, those proteins are in your body. So your body has your immune system. Your body has your mucosal immune system. You know that when you swallow a oral cholera vaccine or oral other vaccines, that helps to develop antibody in your body. So that is due to the response that your body produces against the very whatever pathogen, or it's part of the pathogen inside your body. So think about the bacteria that you have in your body. They are transcribing and translating proteins, producing enormous millions of different types of proteins, and your body is handling that. Because when you are swallowing an oral vaccine, your body is producing some response, antibody response, and that is helpful for you to be protected from one particular infection agent. But think about the millions of bacteria that reside in your gut. Your body is handling that. You are behaving roughly, you are unsocial, you are not very much kind, nice, your other siblings are very nice and kind. Think about that. You are becoming excessively kind of, you know, sensitive. You are sensitive. Someone was trying to make fun and you are very angry. You have blown that, why did you do that? But someone can be even blown and they can be laughing. Like, oh, you don't know that is doing it, like friendly kind of, you know, gesture. But this is how it is taking place. You cannot just define it. But from the signaling point, you can understand that your body, like your body DNA, is producing such proteins, which are, in other words, helping. Some DNA are transcribed into DNA proteins, and that proteins help you digest or utilize things, or maybe someone is stronger, someone is very efficient in utilizing one food, digesting one food, others cannot. So these are the different things that your body DNA is doing. But now we understand that this is not your body DNA, it is bacterial DNA. Not body DNA alone, but combined.
Can we go back to the bacteria profile from the cholera child, like the charts of the the first, yeah, this one.
Oh, this one.
Yeah. So, this is the same one, right? Or
The middle one is
no, this not this one.
This is the diarrheal child. This is one child, day 0, 1, 2, 3 up to day 28. So you see the dilution of the profile DNA.
Oh no, I was thinking
This is
about the
balanced.
antibiotic that you showed, like the Ecology increases and
Oh,
Vibrio.
probably this one.
Yeah, that can.
Yeah, this
Yeah,
one.
here.
Yeah.
So on the right side, on the day 28, there is a like Vibrio cholerae
Band here?
is coming, right?
Here it is.
Yeah,
It is. No, basically this
This should be there.
No, actually, basically this the temporal temperature gradient gel electrophoresis is something that cannot give you the finest resolution of bacterial identification, but it is just closely related bacteria. Maybe the same bacteria belonging to the same group. I mean the
Like genetic
Yeah, this one. Like one band can be one bacteria or 10 to 100 different bacteria, close related bacteria. Who knows? Can be infected. I'm not sure. Could be Vibrio powder. Day 28 means maybe the neighboring people had cholera. Maybe this person was affected again. Yeah? It is likely. But from this kind of study, you cannot say for sure that this band is of Vibrio cholera.
Can I also ask a question about this? So, did you mention that E. coli band is from resistant E. coli or just?
Oh, there's from our subsequent study that we preserved the stool samples.
Yeah, that particular one, is that resistant E. coli?
Resistant E. coli, much resistant.
So when on day 28, do I understand it, since I'm not a microbiologist, so at day 28 it's completely gone?
No, there is the band is there, but the proportion is very less. So I can see the band here. So
they were removed mostly.
This is due to the kind of floral kind of shifting. Basically, during the recovery stage, like the microbial interactions take place. So the bacteria that is not very much likely to be colonizing can also be there, but the native flora, which is likely to be prominent, can be inhibited by that for time being. So maybe something is going on. But can you see a little bit of?
I think you show the calculation later where there are two percentages both.
Yeah. So this is not a. I mean, what I wanted to show you is that as of day 28, the floral restoration recovery is poor. So the number of flora is very less, far less. But in the other slides I have shown, see, yeah. So here you can see, this is the diarrhea children, okay? This is the malnourished children. So diarrheal pattern and malnourished children pattern doesn't change much actually. See, diluted, but compared with the healthy, so more diversity. This proportion is different. Proportion of each of the bacteria can be different. So the diversity, when we analyze the community DNA by genome sequencing, we saw the healthy had more diversity, which is reflected from the TTG slide with many different bands.
I'm afraid I'm going to have to cut off this rich conversation just because our antimicrobial research group has a full day of work ahead of them. But thank you all so much for coming today, and thank you, Dr. Munir, for a really enlightening, interesting presentation. Thank you.